Historically, animal research in neuroscience has exhibited sex bias towards male rodents, meaning that female rodents have been utilized less in this field of research. This does not consider the potential differences in gene expression and pathologies between sexes, such as stress-related neuropsychiatric diseases like post-traumatic stress disorder. Preliminary research has found that the locus ceruleus, a nucleus of norepinephrine neurons, plays an important role in mediating the stress response. Our gene of interest, the mu opioid receptor, plays a role in mediating the stress response. Research has shown that female rodents express lower levels of mu opioid receptors when compared to males. This difference could have implications for how females respond to stressors and possibly can explain behaviors that lead to opioid addiction.
The sub-CD subpopulation of norepinephrine neurons has been shown to be important in the consolidation of stress-induced memories. Studies have shown that stress-induced memory consolidation was inhibited in sub-CD knockout mice. Therefore, mu opioid expression in the sub-CD could play an essential role in stress-induced disorders. Although the Allen-Mainbrow Mouse Brain Atlas shows a low level of mu opioid expression in males, data on expression in females is unavailable. Therefore, we propose to investigate mu opioid expression in the sub-CD region of both males and females. This would allow us to compare expression between male and female mice in order to obtain further insight on the role of sex differences in stress-induced disorders and its potential impact on the pathology of opioid addiction.
Given this line of reasoning, we expect that histological analysis of norepinephrine neurons in the sub-CD will show higher mu opioid expression in male mice than female mice. To test this hypothesis, our aim is to use immunohistochemistry to assess possible sex differential expression in the mu opioid receptor. We'll use tissue that is pre-stained for dopamine beta hydroxylase, or DBH, as marker of norepinephrine neurons, and we'll use antibodies targeting OPRM1 to identify the mu opioid receptor. Microscopy will allow us to compare expression of the receptor in the sub-CD region of male brains versus female brains.
Cellular phenotypes can be an important indicator of both behavior and group-based risks for various diseases. Our proposed research will not only further our knowledge about the norepinephrine system and the mu opioid system, but it will also influence the diagnosis of stress-related diseases, one of the most prevalent in today's society. Also, by choosing to use both male and female mouse models, we can gain an understanding of the sex differences in receptor expression, which could shed light onto the reasons that this disorder is differentially diagnosed between females and males, more so in females. The sub-CD could not only be identified as a target for stress-related diseases, but it could shed light onto its uses as a therapeutic target for other diseases, such as the opioid addiction that has swept the nation in the recent years.